Well, it gives me a great deal of pleasure to introduce Ian Patterson, who is the inaugural Gilbert Stork lecturer. It's a fantastic event for us, which we will have every year because of the generosity of Gilbert Stork and Ako Yamashita, who arranged to endow a lectureship. And so this lectureship will be in the area of synthetic organic chemistry. It will occur once a year. We only require people to give one lecture, but Ian has a lot of stuff to talk about. So he's going to give two lectures today and tomorrow. Tomorrow will be the official award lecture, and this is a photograph of a very nice crystal tetrahedron. I found the shape that I wanted at Tiffany Jewelers. And so each year, a renowned scientist in synthetic organic chemistry will get a crystal tetrahedron as a Stork lecture. As I said, this is the inaugural Gilbert Stork lecture. And so most of my comments about Stork will be tomorrow. Ian is going to make some comments about Stork today. And that way, it won't be the case that I steal some of his stories. Although there are so many Gilbert Stork stories that the likelihood of us overlapping is slim. So here's a picture on the left of Gilbert Stork and on the right of Samuel McIlvaine. And McIlvaine's wife is in the center, okay? All of you know the name McIlvaine very well because of the McIlvaine seminars. McIlvaine was the person who agreed to have Gilbert Stork join his remote research lab. I encourage you to read some of the stories here in this brochure. Stork wanted to go to graduate school at Illinois, so he just headed for Illinois. He wanted to work for Roger Adams, but he didn't apply for graduate school. So he didn't even get to speak to Roger Adams. They stopped him at the outer office and sent him on his way. So he took a bus to Madison, where there was a Roger Adams student, Samuel McIlvaine, and McIlvaine said, okay, you can go in this lab up on the other floor, and McIlvaine only visited that lab once a year. And McIlvaine was surprised to pick up an issue of Jax one day, and there was a single author paper in that by Gilbert Stork, from the remote laboratory one floor up. And McIlvaine, in Stork's words, flipped a little, but not too much. But his response was to move Gilbert down to a lab adjacent to his office where he could keep an eye on him. So, that is Gilbert after he left Wisconsin when he was in his independent career. Here's a picture of Gilbert Stork from a couple of years back at age 90. He still goes into his office, he still works, he still has a total synthesis going. And finally, here is a depiction of Gilbert Stork on the occasion of that 90th birthday celebration. This is a depiction drawn by his son Philip. So Gilbert Stork is a beloved and renowned synthetic organic chemist, and we are very happy to honor him with this seminar. Now, to the issue of our visitor, Ian Patterson has connections to Wisconsin as well, in that he was a PhD student with Ian Fleming. And Ian Fleming spent time in our department as one of the early Gehring professors. And so Ian was a postdoctoral associate with Gilbert Stork in 1989 and 90, and was involved with the total synthesis of 9S-deoxyerythronolide B, a very important milestone in synthetic organic chemistry. Since that time, Ian has become the man in the area of polyketide synthesis. He has developed strategies and methods that solve the various stereochemical problems that present themselves in natural products that are derived from polyketides. And so many of you know the complicated issues of stereotriads and stereotetrads, where you have alternating methyl and hydroxyl substituents off a carbon chain. And much of Ian's work has been to develop methods to solve those problems, and he has synthesized dozens and dozens of complex molecules using those methods. So today he's going to tell us about some of the challenges and discoveries in polyketide synthesis.
Right, thank you very much, Steve. I have to say it's an amazing pleasure to be here. When Steve sent me this email with the invitation letter, I think back at the beginning of this year, to me it was such a surprise. It actually meant more to me than getting a grant proposal funded. So this is such an amazing thing that Gilbert Stork, who of course has got such fond memories of his time in this department and still has, I guess, long-term links with many people on the faculty, that he, together with Ayako, have decided to endow this lectureship series. So for me, yeah, it's great to be here. So let me give you a little bit of an extended introduction to some of the things that Steve has. Oh, I'm going to quit. And then okay, looks like we're in business, which is good. Okay, so this is lecture number 1. And lecture number 1, other than saying a few nice things about Gilbert Stork, will really give you an overview of maybe my philosophy in doing chemical synthesis, and share with you maybe 3 particular projects which we've worked on over the years. Tomorrow will be a little bit different, that what I'm going to do is focus on a single, I have to say, very complex natural product that took us a long time and a great deal of effort to synthesize. Okay, so starting up, here's Gilbert times 3. And here's Gilbert a little bit more recently in his office at Columbia. I think this was back in May this year. And I think it's a very characteristic image of Gilbert, that he's smiling. I think he's sharing a very good joke with Richard Sung and Ayako. Gilbert is very much, let's say, a unique character, and it's very easy to define him to some extent and his contributions to science and its character by his name. Okay, so Gilbert is a giant, and I don't mean anything about a weight problem. Coming from Cambridge and Isaac Newton, we talk about standing on the shoulders of giants. So in terms of organic synthesis, to those of us who do it, Gilbert Stork is easily one of the giants of the field of organic synthesis, particularly in the second half of the 20th century. Gilbert is an inspirational character. So if you talk science with him, if you talk about anything with him, he always has a kind of interesting approach to the particular subject matter and is always very engaging. So as I say, this is characteristic of Gilbert. He has a very good sense of humor and generally is laughing about various things from time to time. And so this particular shot gets him in mid-smile, mid-laughter. In terms of background, he is indeed Belgian. He was born in Brussels on New Year's Eve in 1921. He moved, I think, a few months later to France and was brought up and initially went to school in Paris, and then clearly would like to enjoy a warmer climate and moved to the south of France, and then came over to the US in 1939, initially to Florida, and then, of course, to this department. He's famous for many things, but he really should have more than one named reaction. But the named reaction that he's famous for is the Stork-enamine reaction. But it would be very tempting to put enamines and enolates, because enolates figure very prominently in all of his science. And his favorite functional group, which is also my favorite functional group, is the carbonyl group, and particularly using enolate bond constructions. He's famous for taking radical reactions or radical chemistry out of the hands of physical organic chemists and applying radical reactions, particularly things to controlled cyclizations and elaboration to give complex structures from relatively simple acyclic starter materials. Of course, he's famous for total synthesis. His philosophy in total synthesis is very much a matter of the journey is often more important than the destination. So how you go about the synthesis, you really have to be innovative, you have to problem solve, and if you make the target, that's great, but making the target is not necessarily the #1 objective. He's, of course, done the total synthesis of a whole range of different natural product structures. He is probably the first person who thought seriously about control of stereochemistry in total synthesis, and particularly exploiting things like stereospecific reactions. His training is second to none. So the Stork School has turned out of the order of about 120 graduate students and excess of 200, probably up to about 250 postdocs. And roughly about a third of that number have ended up going into academic careers. And a good number of them are still in academic careers, which is quite impressive. And all over the world, I should stress. Okay, organic chemistry, well, Stork is one of the pioneers of sort of innovative organic synthesis and new reaction development. So as well as stereo control, regio control is important as well. And in terms of his character, for those who know him well, he's one of the kindest and most considerate people and has amazing qualities in terms of his personality. And that's, of course, reflected in his very generously endowing this lectureship here at Madison. Okay, so this is for me a little bit like a trip down memory lane. So here's Gilbert again, back in Madison. And hopefully you can read this, because I'm not going to read it out. It has something to do with him being very concerned that he was going to blow up the chemistry department here at Madison. Fortunately, he was unsuccessful. However, rather than telling you the story, I give you all, those of you who have not read this already, some light recommended reading. So this article by Jeff Seaman in Angevanta in 2012, celebrating Gilbert's 90th birthday, essentially describes many of Gilbert's adventures based on his words and the musings of his friends. So this is just one example, and there's many more. Very entertaining. So Gilbert, via Harvard, ended up being professor at Columbia. Interestingly, I just sort of noticed it's from 1953, so that's actually 1 year before I was born. So it's just amazing that he's put so much of his energies and enthusiasm into, let's say, building up that organic department at Columbia. So one thing which I will read out here, the Stork reaction, 1954, originated with wondering what might be going on in nature at a very primitive level. How does nature manage to make carbon-carbon bonds? And that was back in 1954. So at that time, he, to some extent, again, pioneered the idea of bio-inspired synthesis, thinking about the polyene cyclization to establish complex terpene scaffolds as in steroid systems, how one can use enamines, and of course that forms the foundation of what we talk about today as organic catalysis. So definitely this is required reading. So let me go back to my time in the Stork group. First of all, Steve, I have to correct you. It was 1979 to 80. I think you made me 10 years younger, which is very nice of you. OK, so this guy here looks as if he's, first of all, very well equipped in terms of hair to survive the New York winter. So at that time, I guess I was a bit of a razor dodger, which had the advantage that it kept me warm over winter, and it saved me time in the morning shaving so I could get into the lab just a few minutes earlier. So I look very happy here, certainly very relaxed, and that was probably one of the happiest times in my career professionally, that one could just enjoy the chemistry, interact with Gilbert, and interact with other people in the group. And it was very much an international group, so I made certainly long-term friends. And so here's some of those long-term friends. So this is 80 to 90, so 10 years on. So my beard is a little bit shorter. I've moved up to having a tie, but I actually feel uncomfortable with that tie, so it's a little bit not fully tied up. And so this guy here is a famous Japanese chemist, Eiichi Nakamura, at Tokyo. He's a very good friend. He worked in the same lab as me. And coincidentally, in terms of arranging this visit here to Madison, he's tomorrow, while I'm here, he's in Cambridge, so we're kind of missing each other, but never mind. And this is Takahashi, who's an amazingly jovial Japanese chemist. So this is Tokyo 1990, and then what we're talking about is group reunions. So having an international group is great, so that when Gilbert travels around the world, invariably members of that group will get together and we'll have a nice dinner to celebrate Gilbert's presence. So this is now a little bit later in Manchester, and I guess I'm at the back there, and several other people here. Some of you might know Verinda Agaval at Bristol is hiding here at the back. OK, so fast forward. So early this year, Gilbert with Richard. So it's really nice, and I would love to be there as well. OK, so Gilbert is a remarkable person, and I've been so fortunate to have had the opportunity of working with him. And I have to say that he has been inspirational with the kind of chemistry that I've tackled, because I've always thought, yeah, it would be nice to be able to make these complex natural products, ideally trying to make them in a reasonably efficient manner, and where it's important to consider, is it feasible at all to think about transferring such an academic type synthesis into an industrial environment. So I have two slides here, and I realize you may not actually be able to see or make out the structures, but we'll have a go. Right, so these are small molecules, so therefore they are small. You don't really see what they are. So in my office and in the lab, I have this and another poster, which really is to remind me of the target structures we've worked on in the group. So they're primarily polyketide, which means that they're very rich in stereochemistry, highly oxygenated, and in most cases, the large majority of cases, have significant biological activity. In fact, some of them may actually be used therapeutically. So what are the perhaps most important ones here, other than independently coming up with our own synthesis of erythromycin, is when we got roughly through to the middle here, a compound which we worked on, discodermalide, which was way back around about 1999-2000. Even before we finished our first synthesis, we were contacted by Novartis in Basel, who wanted to develop discodermalide as a new anti-cancer drug. And since it comes from a sponge, they needed a synthesis. So we worked with them. And for me, it was incredible, the level of resource they would put into producing initially, I think, 60 grams for phase 1 clinical trials. And then even though they didn't do the last 2 steps, they were aiming to make 1 kilogram for phase 2 clinical trials. So molecules of this complexity can be made in industry, provided, importantly, they're prepared to put the resource into the project. Okay, so that's slide 1, slide 2. So this is more recent. This is going on from about 2002. So it's roughly maybe the last 10, 12 years. So what I'm going to talk about today is our work initially on, where is it gone? Oh, it's up the top. Something called sporangion. So this sort of spiroacetal polyene with a side chain here. Then I'm going to move on to discuss something which we don't normally do, which is structural elucidation before we actually tackle the total synthesis. So this is leodermatolide. And then if you're still with me and haven't fallen asleep or sneaked out the back, I can actually announce that we've recently completed a total synthesis of aplonine A and aplonine D. Okay, so let's kick off with myxobacteria. So myxobacteria for organic chemists really became important in the context of the apothalones. So the apothalones, I guess synthetic chemists were working on the apothalones about 15 to 20 years ago. And apothalone comes from myxobacteria. And the one which actually has gone and been approved and is in the clinic now for advanced metastatic breast cancer is Exempra, which is of the lactam analog of this particular macrolide. So the group of Höfle and Reichenbach in Germany at Braunschweig, they put out lots of patents, primarily in the 1990s, on complex natural products isolated from myxobacteria. And most of those had promising anti-cancer activity. However, looking at those patents, invariably, it's just a planar structure. And most of these molecules have many stereocenters. So it would take a long time before synthetic chemists actually had the courage to perhaps consider the synthesis of these structures. So one of them, which we finished last year, is a molecule called rhizopodin, which is a symmetrical macrodialide. So you might consider this a kind of lardy type structure that, I mean, apothalone could probably fit in the middle here with no difficulty. So with our synthesis, it was really quite frustrating that when you tackle these molecules, importantly, of course, you have to think about getting the connectivity correct, putting in the functional groups, putting in the stereochemistry, and at least we're not smart enough to do these syntheses without protecting groups. So a critical issue at the end of these syntheses is, can you get the protecting groups off cleanly to give the natural product? So in the case of rhizopodin, this was very frustrating, because the first 2 times we got to the finishing line, we could not get those protected groups off without destroying the molecule. And it was only ultimately on the sort of 3rd attempt where we had, so it's a kind of a tetraol, so alcohol here, alcohol here, alcohol here, alcohol here, that we had the right combination of silyl protecting groups that we could get them off to produce rhizopodin. So the reason I say that is because with chivososol, we actually have a synthesis of a protected version of chivososol, which has got silyl protecting groups and an acetonide. And I feel that if I talk about that synthesis, it really is going to be then a situation that, having talked about the synthesis of a protected version of chivososol, we can't actually get those protecting groups off. So what I'd rather share with you is a synthesis which I think quite nicely illustrates the strategies that we follow in my group, particularly aspects of using, say, substrate-based induction, a little bit of reagent control, perhaps where there were no real issues of protecting groups, but there was a certain fearful-looking side chain here. There's this pentaene, which has really got alternating ZE, ZE, Z double bonds, which really don't look happy at all. In fact, it's surprising it is actually a natural product. Okay, so in terms of history on this project, the first paper which actually showed any stereochemistry at all came out in 2005 and emphasized that these compounds actually have significant activity. And if one degrades this, so an unusual way at the time of doing a degradation, instead of ozonolysis, they actually do a kind of cross-metathesis with ethylene. So this would chop out the side chain to give this particular diene. And this truncated version of spirangium A has about 1/10 of the activity of the natural product, so it's retained. So this was characterized by NMR and X-ray, but the X-ray analysis did not give an absolute configuration. So what we decided to do is firstly to go after this to determine, well, what is the absolute configuration? And then when we figured that out, to go on and try and make the natural product. Okay, so synthetic planning. So spiroacetal, one thinks about opening this up to a ketodiol. So open up the ketodiol and protect this hydroxyl with that hydroxyl, that hydroxyl with that hydroxyl as acetonides. Chop off this pentaene side chain, because this is so delicate that really you would have to put it in right at the end of the synthesis. Because if it sees any light, anything acidic, those double bonds are going to start isomerizing. So this is going to go in late. So we have this kind of acyclic equivalent of this. And also we'd have to do this carbon-carbon bond-forming reaction to put in this trisubstitute double bond. So the beauty of this is if you look at these polyketides long enough, you often start to see symmetry and possibility then of further simplifying the synthesis. So if we cleave across here and look, we have 5 contiguous stereocenters, 5 contiguous stereocenters here. The main difference between what's on the left-hand side and what's on the right-hand side is here there's a syn relationship and here's an anti-relationship. So if we cleave back further and get back to this stereotetrad, that particular stereotetrad should, in principle, be able to give us the left-hand side and the right-hand side. And, of course, imagine this being a carbonyl, so disconnect by aldol to this chiral ketone, which comes from Roche ester and methacrylene. Now, the attractive thing about this analysis is that we actually had developed a very general approach to making stereopentads where, depending on the conditions of the aldol reaction, the reduction, or the hydration of the double bond, we could actually access with very high selectivity any of the 32 possible stereoisomers. However, it turns out that this is one of the easiest ones to get. Okay, so here's the starting out the synthesis. And so the last time I was here in Madison, I had a very enjoyable talk with Howie Zimmerman. And so it's rather sad to come back here and knowing that Howie relatively recently passed away. But here, this is a great opportunity to me to emphasize, of course, one of Howard's early contributions, which is the Zimmermann-Traxler transition state. So this is actually a very special Zimmermann-Traxler transition state. It's not chair topology, it's more of a twist boat topology. And it's actually a sort of bicyclic type transition state where this PMB ether oxygen is forming a hydrogen bond through to this formyl hydrogen of the complex aldehyde. As a consequence of this highly organized transition state, which is also determined by DFT calculations, gives the aldol product through enolization to the E-boron enolate this particular adduct with excellent stereo control. And this could be done on, if you really wanted to do 50 grams, 100 grams a kilogram. So a samarium catalyzed Tishenko type reduction gives you this 1,3-diol. Now we have our common intermediate. And we knew from earlier studies that in these systems where you have an allylic alcohol, and if you use a sterically undemanding hydroborating agent like borane, then one can actually get the syn selectivity for that methyl group versus the hydroxyl group through this particular transition state. So a little bit of manipulation goes through to the aldehyde. And then for the other piece, so here's the common intermediate, tie it up, go through to the iodide, now using some of Andy Meyer's auxiliary chemistry. So this glycolate aldol or glycolate alkylation works very nicely to make this carbon-carbon bond and control this hydroxyl-bearing stereocenter, and cleave this off with methyl lithium to give the ketone and sialate. So at this point, we can make the fragments, but one of the most challenging issues, often in complex polyketide construction, is joining the fragments together, particularly if you're also going to control stereochemistry. And while there's a lot of literature out there, and maybe you have a confidence level that just maybe you'll get the right stereochemistry, often you encounter completely new situations. So this is actually a new situation. So this methyl ketone with alpha oxygenated stereocenter and this alpha methyl beta oxygen aldehyde uniting together in aldol coupling, that what one would predict, and actually turns out to be true, is that one would expect to get the syn, which is corresponding to Falcon and control, and with lithium or dicyclohexyl boron, in fact, you get very largely the syn, which is the undesired adduct. So in this situation here, we do need to use a chiral reagent to help us to overturn that substrate-based induction. It has to overturn it, so the selectivity you're going to get out may well just be moderate. So we're actually reasonably happy, but we could be happier still if it's higher. But 2.2 to 1 with an IPC aldol reaction to give us the desired adduct was actually not too bad at that stage. Okay, so going on, we methylated. Now we can make the spiroacetal. So these hydroxyls essentially sort themselves out. And also we remove the test. So this thing just zips up to give 2 spiroacetals. We can now separate the epimers at C23. This is the one that we require. And all of this NOE analysis establishes that. However, we now reached a problem. So we tried very hard to take that intermediate forward to give the sort of the dying degradation fragment of sporangium. But the difficulty is when we I think put silyl protecting groups on, remove the PMB ether, convert that to the iodide, coming in with some kind of alkenyl nucleophile to displace the iodide. When you have the spiroacetal and maybe a silyl ether here, it's really very crowded, that sterically that totally shuts down that displacement. So we couldn't go forward. So if you can't go forward, you have to go back and reorder some of the steps. So reordering the steps, so this is the original approach where we went this way. Fortunately, all it requires is you put the iodide in earlier, do a displacement, copper catalyzed displacement of the vinyl lithium here, actually works extremely well. Then knock off this and give us the aldehyde. So now we can repeat the aldol addition, methylate, remove protecting groups, and gives us this with this side chain built up and the correct configuration. So we're in better shape. And now acylation, and when you have a silyl group here and use a bulky hydroborating agent, which is really based on the work of Clark Still, then reliably you get an anti-relationship of the methyl group to the silyl ether next door. So substrate control again gives you this and goes through the aldehyde. Now from our work on discodermalide, which has sort of dying terminus, we thought this allyl chromium reagent should work in this case. But in this case, because of the bulk of this spiroacetal, it actually gives a mixture where you get reaction at the desired end of the allyl chromium, followed by Paterson elimination. But you get competing reaction in the allylic sense, which gives you this as well. And this is actually the major product. So again, an unexpected difficulty, and we have to try and come up with a solution to that. Well, of course, one can go from allyl chromium to some other allyl metal species, and allyl boron works quite nicely in this case. So the corresponding allyl boron reagent now reliably gives you this, and you can do the Paterson elimination to give the diene, remove the silyl groups, gives you this, compare the rotation with that reported for the degraded compound, and we're actually in the correct enantiomeric series. Okay, so I realize this is sort of Freak Fest, Halloween, so I had to put something inappropriate. So this is literally, you have to do the final steps of this synthesis in the dark, avoid all possible light. So one thinks about the story of Dracula. And so in doing this, importantly, this aldehyde, combine it with a Stork-Wittig olefination, gives the Z vinyl iodide. Remove the silyl protecting groups, gives this. And then actually a very difficult Stille coupling. First of all, you have to build this up with the EZ tetraene geometry and hold that geometry, which means you have to do that in the dark as well. Followed up now by the Stille coupling with this vinyl iodide, all done in sort of foil-wrapped flasks, light switched off, minimal purification, go down to the NMR machine, run the crude NMR and hope that the pentiene's there as it should be. And with the appropriate precautions, we could actually achieve that. So this is literally a nightmare, this sort of ZE, ZE, Z. So we made the methyl ester, which is actually a little bit more stable than the natural product, and hydrolyzed that to give the acid. So we made the natural product, but I think after probably a few days, we'd made many natural products because there is spangen A, B, C, D, E, F, etc. And the only real difference is that those congeners have different alkene geometries. So maybe this is sort of my attempt at doing diversity-oriented synthesis. Well, you sort of make a mixture. Okay, so we actually completed spangen A. And I think one thing I should stress here is, okay, we're all conscious, those that do, let's say, natural product synthesis, that based on Barry Trost, who of course used to be in this department, we have to go for atom economy. Based on Paul Wender, we have to go for step economy. We have to go for redox economy. So in my group, I go for student economy. So this particular synthesis was essentially carried out by one PhD student over 2 years. And prior to that, she did a synthesis of another natural product called dolistatin 19. So she was very talented, Alison Finlay. Okay, so probably go back. So this is kind of an overview of the synthesis. So this thing could be made tens of grams, hundreds of grams if you really wanted to. So you sort of diverge, converge to make this. 5 steps gives this aldehyde. We made this diene. We made spangen A. These compounds are actually pretty potent, but not chemically stable. If we really wanted to pursue this, then of course we could redesign at least the side chain into something which had a bit more chemical stability and maybe explore the chemical biology of these systems. Okay, so you've been very patient, and this is the commercial break. Maybe. Okay, so Cambridge is a fantastic place. It's a very old town, and the university is very old, about 800 years. And everything tends to be quite slow there, so this is still an important means of transport, so punting along merrily to get from A to B. Chemistry department is certainly not here. Chemistry department is somewhere over here. Okay, so this is where the work is done or close by to King's College. But as I say, sort of introduction to the second topic. So in terms of water, this is the River Cam, and I'm sure there's no, well, there probably are some kind of living organisms there, but you wouldn't want to ingest them. I mean, maybe there are some natural products, I'm not sure. Okay, so this is pretty much the stimulus for next topic, which is thinking about marine natural products in particular as potential anti-cancer agents. So one of Gilbert Stork's, perhaps, I don't know if it's famous or infamous targets, it certainly give him a lot of fun and I'm sure frustration together with a yakko, is Taxol. So Taxol, of course, is very important as an anti-cancer drug and is still in widespread use together with semi-synthetic version, Taxotere. But there are problems in the clinic of resistance, both acquired and intrinsic resistance. And so as a consequence, of course, that was the reason for the development of Exempra as a version of apothalones. But there's several marine natural products, discodermalide I mentioned earlier, lolomalide, pluricide, and dictystatin, that share the same microtubule stabilizing activity as Taxol, so the same mechanism of action, structurally very different, and the way they bind into tubulin are different from Taxol as well, although some of them certainly have overlapping binding site. Some of them have a very distinct binding site. So there's a possibility that these things could actually be developed as anti-cancer drugs in their own right. So that's why Novartis got interested in discodermalide. We subsequently got interested in dictystatin, which was originally isolated by Bob Pettit and then re-isolated by Amy Wright, who's at Harbor Branch Oceanographic Institute in Florida. And the stereochemistry wasn't known, or at least Pettit had proposed some stereochemistry, but some centers were undefined. So we managed to get a sample of that compound from Amy, and based on extensive NMR analysis, we actually came up with this particular relative configuration here. And the absolute configuration was kind of implied by comparing it with discodermalide. So that was really our first opportunity to do, let's say, basic structure elucidation in terms of stereochemistry. And we got interested in another molecule that Amy Wright had isolated from a different sponge, probably now about 5 years ago. And this is something which goes by the name of leordomatilide. So leordomatilide, okay, certainly has potency in the same range as dictystatin and discodermalide, so it's low nanomolar against a range of cancer cell lines. It appears to be doing something with tubulin, but the details of how it interacts with tubulin is not known, but it's certainly distinct from other tubulin targeting anti-cancer agents. However, the structure at the time was not known, so in this patent back in 2009, Amy just really gave a planar structure. So if you're going to tackle it as a synthetic project, we didn't really want to almost like randomly make stereoisomers and hope we got lucky. We wanted to try and determine what is the likely stereochemistry first and then consider that as the synthetic target. So we had to do various sort of, I guess, reasonably straightforward NMR analysis, at least on the kind of things that natural products isolation people do. What is more demanding is to do this Murata-J-based configuration analysis, where you have to pick out the heteronuclear C13 proton coupling constants, which requires more sophisticated NMR experiments and certainly a lot more time. Lots of NOE experiments combined with molecular modeling. And then what was particularly useful here was based on using some of my former students, now colleagues, Jonathan Goodman's method, which is a computational NMR-based technique to calculate the chemical shifts for both C13 and protons for candidate stereoisomers. and then compare that with the experimental data and come out with a probability value as to whether that is the most likely structure. So we used this. What was unsuccessful is determining the configuration. So this then is this sort of macrocyclic system with a little bit like spangen. So now it's a ZZ diene and an EE diene out here, and an isolated trisubstitute double bond, and a carbamate there. And progressively then, we could determine through this analysis, the relative stereochemistry of the macrocycle is shown here, which then was based on NOE analysis as well as the DP4 probability method. So we had some confidence that we actually had this stereochemistry here, and here's just some modeling pictures of it. So we had 4 possibilities because we could not, from the Moshe analysis, determine the absolute configuration of the macrocycle. And we couldn't relay stereochemical information from the macrocycle into the side chain, which is this hydroxy delta lactone. So really there's 2 possibilities here, 2 possibilities here, so 4 possible stereoisomers. And as it turns out, when we put out this paper, we, to some extent, tossed a coin and said, maybe it has this configuration, so a 25% chance, and I wish I'd bet some money on it because that 25% turned out to be the correct one. Okay, so what we went about was, this is sort of, let's say, the first generation approach. First generation approach, which is described Orglet 2011, we could build up this system here. We had some difficulty putting the carbamate in on the correct oxygen. And we had some real difficulty here. So this vinyl bromide, obviously what we intend to do is some kind of palladium catalyzed coupling to build in the side chain. We made this, we managed to destroy it. We could not get any cross coupling to build in the side chain. So on the positive side, we did sort of NMR comparison of this core versus the natural product, and so proton comparison, carbon comparison, NOE, and coupling constant comparison. So we had confidence that that indeed was the relative configuration of the macrocycle, but didn't know whether it was the correct absolute configuration. So we went back and thought, well, how do we solve this problem? Well, we solved the problem by, of course, of building in this side chain earlier. And so this is now the second attempt. So leodermatolide, we have to put this carbamate on. Maybe we could be adventurous. So we're not going to have differential protection here with a 1,3 diol. And build it up then by 3 palladium catalyzed cross-coupling reactions, a Suzuki, a Staley, and a Heck. And at the same time, we of course like to showcase our chemistry. So here's some of our a different kind of aldol construction based on a chiral ethyl ketone derived from lactic acid, either this enantiomer or this enantiomer. So here's our analysis. We're going to make this fragment, and essentially this fragment, join these 2 bits together to give that, and elaborate that to give that, connect this to this. Cross your fingers, you can actually close the macrocyclic lactone, because in new situations, one can never have complete confidence that you can actually close the ring, so it's sort of a big unknown. And then if we've got any protecting groups, of course, can we get those protecting groups off? Okay, so C1 to C11, which is this bit down here, just to go through it quickly. So Roche ester derived Weinreb amide, go to the ketone, make the vinyl triphylate Suzuki coupling, builds in now the trisubstitute double bond. Through this aldehyde, doing the first boron aldol with the lactate derived ethyl ketone, gives you essentially only 1 adduct, because this is now a sort of matched combination of the alpha chiral aldehyde with the induction from the enolate. And then this is elaborated by alkyne addition to this cleavage of the diol back to the enone here. Conjugate addition of iodide under acidic conditions, we've sort of optimized the conditions now to give about 15 to 1, Z to E to give this enone here. This is now a good substrate for an Evans-Saxena reduction to give the 1,3 antidiol. So we don't differentiate these hydroxyls, but we do actually put a protecting group on, which is the acetonide, and take the iodide through to the stannine. So for the other piece, so this is first of all to make the side chain. Again, a boron aldol sets this stereochemistry. Allyl-Grignard addition into the carbonyl gives you now this sort of deconjugated enone. We can now use a Mukayama aldol and take advantage of that stereocenter and to some extent that stereocenter to get high facial selectivity through this open transition state to give now, after acidification, the corresponding lactone with the desired configuration at that center. This vinyl iodide is made from this ethyl ketone, and now, while in our first generation route we were trying to do things like Stille couplings and Suzuki couplings, we find here that simply an allyl group on the lactone and a Heck reaction works very nicely to build in now this E-diene system. We cleave to the aldehyde, another Gilbert Stork Wittig, which we like very much, gives us the Z vinyl iodide. Okay, so moving on, we can do the Stille coupling. That works nicely to give the ZZ diene. Remove most of the protected groups, at least the silyl protecting groups, and adjust oxidation state to the acid. Unfortunately, this thing will macrolactonize. It'll either macrolactonize this way as shown, or you can actually take the acetonide off first, and then you have 1, 2, 3, 4, so of a tetraol, but that one is nicely set up only to lactonize onto that secondary hydroxyl. So it looks reasonably good so far. Something bad has to happen. So the something bad that has to happen is, okay, if you naively look at this 1,3-diol, you think C9 with sp2 carbon next to it and just an alpha methyl is in a less hindered environment to this position. So if you derivatize this, such as by esterification or even silylation, it should go on the desired hydroxyl. And silylation certainly goes on the desired hydroxyl. But the most normal reagent system for putting in a carbamate is trichloromethyl isocyanate. And when we did that, we got a rather disappointing result that the major product was on C7, so it was the wrong carbamate regioisomer. We got a little bit of what we wanted, and the remaining material was the biscarbamate. So we were in the situation then of, it's a reasonable route to this 1,3-diol. How do we rescue the synthesis? So we had to do a little bit of silylation chemistry. So if you put TMS on both of those, the more labile one to come off with PPTS is C9. Then we can put the carbamate on and then remove the remaining TMS. So that sort of solves the problem, and we actually made leodermatolide. And that correlates exactly by NMR and specific rotation with the natural product. But comparing particularly the proton NMR of this versus this diastereomeric side chain is very difficult. We were planning to make this, but we actually got scooped by Fürstner in Germany because he'd seen our paper, decided, well, he wanted to make this natural product as well. So he made this and actually found that this particular diastereomer, there are really very subtle differences in the proton spectrum relative to the natural product. So in fact, this must be leodermatolide. So we finished our synthesis. We can, of course, make analogs. We can hopefully scale up. So that brings me on to the third topic. And this is one of those natural products that I've worked on now for so many years that's become a good friend. But as well as being a good friend, it's a bit of an enemy in that if you really hope to finish the synthesis, you need to have certain things work out for you. And in our PhD system in the UK, in the early days, which is actually when we started this, PhD program was strictly 3 years. So you had to have written up, submitted your thesis within 3 years. So that's kind of tough to put a student on this. More recently, it's 4 years, so maybe the student can get a little bit further in the synthesis and maybe get close to finishing. So this is one of these projects which I perhaps would describe as a linear synthesis. And what that means to me is usually, at any one time, I've only got a single student working on it. And they, of course, have got to make all the fragments, build up stocks, and explore sort of the new steps and get closer and closer to the target. But there were times in that period that it was sort of fallow time, so no one was working on the project. And it was only when I sort of forgotten why we'd stopped working on the project and what some of the problems were, I thought, I'm going to put a new student on it. I'm sure I'm all enthusiastic we're going to make it this time. So aplonine A, B, C, D are, I think, very interesting marine natural products isolated way back almost like 20 years ago from this creature here, which is a Japanese sea hare, which goes by the name of Aplysia curidae. The sea hare doesn't make it. It comes from its diet. It sort of feeds on algae. And so the producing organism most likely is cyanobacteria, but that cyanobacteria has not actually been identified as to which one actually produces aplonines. So these compounds, and I only found out this recently, have been tested, at least aplonine A, in the NCI 60 cell line panel. And these are extremely potent or extraordinarily potent. So typically sort of low picomolar activity in almost all of those cell lines, and it's comparable activity to the spongistatins. However, the only one that's been tested is aplonine A. So aplonine A, which is the one at the top here, in terms of structure, so a macrocyclic lactone, a very highly substituted side chain ending in this vinyl formamide, and has an amino acid here, which in this case is dimethylalanine. And here, very interestingly, is trimethylserine. So if you have the trimethylserine residue there, this is an extremely potent compound. If you don't have it, so it's the free alcohol, it's about 1,000 times less active, so low nanomolar versus low picomolar. So in terms of pharmacophore, that trimethylserine is critical. And for a long time, no one really knew what it was doing. Because these compounds, well, first of all, aplonine A has actually been tested in vivo, and it's very promising in terms of inhibiting tumor growth and extending life, at least in mice. And so just last year, the mechanism of action has been revealed that it's not just targeting actin, but what happens is that it's sort of actin depolymerizing agent. So aplonine A actually forms a complex first with actin, and that complex will then inhibit tubulin polymerization. So it has a dual target, actin and tubulin. So in terms of a possible anti-cancer application, this really sort of ticks two very big boxes, something which will go after actin as well as tubulin. So supply is a real problem. There's only one total synthesis, and that was quite some time ago, and so it's more than 45 steps in the longest linear. The reason, to some extent, why it's such a long synthesis is because the molecule is totally covered with different protecting groups, and it becomes, one has to put each of those protecting groups on in a step, and then get them off at the right stage one by one to put in the appropriate functionality. So we really wanted to be a little bit more adventurous than that. And first of all, in terms of comparison with other actin-binding macrolides, so I mentioned this before, rhizopodin, which is a dimer. So these sort of characteristic side chains with this kind of stereochemistry methylation pattern, vinyl formamide, are sort of shared with several other compounds which have the same actin binding mechanism and filament severing activity, but the aplonine A and D are unique because of this trimethylserine, which then also binds to tubulin. Okay, so there's some useful crystal structure information of aplonine bound into actin, and one can establish how much of that structure is important for binding. So in this cavity between subdomain 1 and subdomain 3, kind of hydrophobic cavity, it's largely the side chain which is fitting in first, this is sort of the primary binding event, and then the macrocycle, the secondary binding event. But this trimethylserine group in the crystal structure is really doing nothing because it's sticking out in space, and therefore it's available to interacting with tubulin. So one can actually make simplified versions of actin, or of aplonine, with the idea that, well, maybe you don't need the whole structure. In fact, that's true. So you can make this, which has not got the macrocycle, this, which has not got the macrocycle. And in terms of functionality, these are both pretty active actin depolymerizing agents, but have low cytotoxicity. And we actually have crystal structure data of these things bound into actin, and of course they share pretty much the same position as the natural product and also other natural products like radiospongylide. Okay, so let's look at our strategy, and the strategy has evolved over time based on disappointments, so dead ends, issues of functional group, conversion problems, many, many other things. So, but overall what we want to do is to simplify it at all possible and maybe not go for so much sort of differential protection of the hydroxyl groups, and let the final compound, which is going to be, okay, multiple hydroxyl groups, see if we can actually steer the appropriate amino acid in to the correct hydroxyl groups just based on substrate bias. Okay, so if we cleave off the side chain, this side chain, particularly with the acetate, is probably a little bit delicate, so we'll put it in late. We want to make this macrocycle truncated side chain with an aldehyde, and that then gives us 3 fragments. This fragment here, which is a side chain with the phenylformamide, this middle piece, which has got stereocenter, stereocenter, stereotetrad, and then this piece here, which is really just a stereotetrad with a diene, which is going to become the dienoate. And we can do cleavage there to go from the iodide to establish this for coupling by HWE. Okay, so first fragment, synthesis. Okay, this is not commercially available. You have to make it. It's maybe about 3 steps from commercially available. Chiral ketone, boron aldol, through that bicyclic transition state gives you really good selectivity for this adduct here. It can be done on scale. Samarium catalyzed reduction again, which is the antisense, or an Evans-Dushenko. And now actually a very interesting isomerization reaction of this inoate with triphenylphosphine and phenol gives you the EE dienoate. Knock this off, cyclic protecting group, which means that at the end of the synthesis we have to differentiate this hydroxyl group from this hydroxyl group. Take this through then to the corresponding iodide, do this sort of dianion chemistry, which I seem to remember was introduced by Paul Greco. So this phosphonate ketone, if you deprotonate twice, then the more nucleophilic carbon is this one here, so one can do an alkylation. So we'll make this, the piece that we need to connect this to by an HWE is this one here. And now some chemistry which was developed in my group quite some time ago by Rich Tillier, who's a postdoc in my group, who now is actually a very senior person in Merck, who I think is worldwide head of drug discovery and preclinical science. So it's sort of really nice to have people in my group who end up doing so well in industry as well as academia. Okay, so the tin aldol gives you now quite different stereochemistry through, of course, quite a different transition state, reduction, 1,3-diol. Again, we could have played around with differential protection, but we would rather sort of gamble that maybe we can sort it out later. So cyclic protecting group, silane, HWE, this barium hydroxide, again, sort of introduced to solve this problem, gives us the E-enone. Chiral reduction here sets that stereocenter and through to the aldehyde. Another of these HWE, so it's a very nice way of, based on coupling together relatively complex aldehydes with ketophosphonates, in this case, set this E trisubstituted double bond geometry, again, an asymmetric reduction, methyl ether, and take this through to this diene acid. So here we're starting to gamble. What we'd hope for is certainly in an intermolecular sense, this hydroxyl looks more accessible than this one, which is sort of between these two methyl groups and got TIPS ether, which I'm sure is blocking it. But when it's intramolecular, all bets are off because you're going to be controlled by any kind of conformational bias of the tether between the functional groups. And so initially, we do a lactonization under Yamaguchi conditions, and it gives us pretty much exclusively the wrong ring size. So that's a rather sad event, except knowing that you can do a transesterification. So if you lose titanium isopropoxide as a very mild Lewis acid, you can go from the 26-membered ring size to the desired 24-membered ring size, about 1.6 to 1. You don't lose any material, but of course you have to do this 2 or 3 times, so we can actually build up stocks of the desired ring size. We're now actually trying to work on improving this step in the synthesis so that maybe we do actually put a protecting group here. Okay, so the side chain is the smaller of the fragments, which is probably more of one of the toughest pieces to make. And the way we made it was, again, a sort of tin aldol reaction. Again, this samarium diiodide mediated reduction. Sialate, remove protecting groups to give a diol, and then a double Swerin oxidation gives the ketoaldehyde. Now we introduce this Wittig procedure. So this is, again, a transformation that was required for this synthesis to go forward. So we developed this Wittig procedure, which is kind of nice because it's high yielding. The downside is it gives the wrong geometry of the double bond. But that's not necessarily a dead end, because you can use isomerization of double bonds. And so iodine conditions will actually isomerize that to give the desired E-vinyl formamide. And this is a much better procedure for constructing vinyl formamides in at least, well, this is sort of a relatively small piece compared to kind of more classical approach, which is condensing N-methyl formamide with an aldehyde under acidic condition, which of course necessarily causes degradation. Okay, so we can make this piece, connect them together. So first of all, lop off protecting groups, go through to this 1, 2, 3 tris tes ether aldehyde. And this is now perhaps the most difficult carbon-carbon bond forming step in the synthesis. Initially, we tried to do it by an HWE. It didn't work at all. All we got was an elimination of the aldehyde. We tried various things, but of course, it didn't take us long before we tried boron. Boron works really nicely. So chemoselectively, you enolize the ketone in the presence of the acetate. This thing will add into the aldehyde. You actually get pretty good control of the configuration there. So in the next step, chemoselectively, we can reduce, under Burgess elimination conditions, the beta hydroxy there to give the enone without eliminating the acetate. And then this sort of copper hydride type reduction takes the enone to the saturated ketone without touching the dienoate. So we get through to this. So at this point, we're actually getting close to our target. And what required was screening various reducing agents to control the configuration of C29 stereocenter. And then we've only got one alcohol, so at least at this point, there's no doubt that we'll esterify on the right oxygen, assuming, of course, the esterification works. So with dimethylalanine, it does indeed put in this ester. And then finally, remove the silane protecting groups, gives us aplonine C, which we reported last year. So aplonine C is the one which is nanomolar activity, which just has maybe the shared mechanism of action of sort of actin binding and filament severing. The one we really want to get is this one here, which is aplonine A or aplonine D. So over the last couple of months, I've encouraged one very good student in my group. Again, I have the benefit of getting some really good students in my group. And so Alison Finlay is a good example. Now I have someone, Simon Williams. And Simon has had to repeat quite a bit of that synthesis and build up stocks of this. So of course, with this, you can do the same thing again, stick in the dimethylalanine or dimethylglycine. And dimethylglycine, which now where R1 equals H, almost certainly is a new aplonine. It's in there somewhere, it just hasn't been isolated. So now we're in the situation, we have a trial, and we need to do a site-selective acylation to put the trimethylserine on this position. And on paper, it looks, well, yeah, fine, we'll do that. But in practice, of course, there's a big risk that you get competing acylation on the wrong hydroxyl, or you let the reaction go too far and you have bis ester. So if you're not greedy and only take it through to maybe about 60–70% conversion, you get exclusively acylation on the correct oxygen here, and that now converts aplonine C into aplonine A, and this one, aplonine X, I've described it, into aplonine D. So we've now completed the synthesis of A, D, X, and C. And what we'd like to do now is, of course, make a useful amount, but perhaps not just make a useful amount, because in the last 2 or 3 slides, well, first of all, I should mention where these things come from. So this C here, to isolate 2 milligrams of aplonine D, which is actually one of the very active compounds, requires more than 300 kilograms of the C here. I don't know how many, let's say, I don't know what C hair typically weighs, but maybe 100 grams or so. So that's about 3,000 of these things, which you then homogenize, which I guess is you put in a food processor, and then you extract away with methanol. You do all sorts of kind of solvent partitions. You do very elaborate chromatography. You do HPLC, et cetera, really laborious, and certainly not sustainable. So now we actually have, I think, a sustainable synthesis of these particular compounds. So what I'd like to finish with is what I've kind of highlighted here is nature, of course, produces extraordinarily potent and selective compounds that probably in the natural environment have a well-defined target and a function. And of course, we are trying to think, well, maybe these are good for drug discovery, so it's doing something quite different. But they are very potent and have great promise. And where I think they have great promise in particular is in the area of antibody-drug conjugates, ADCs. And now many of you will be aware there's 2 of these compounds that have been approved, Kadcyla and Adcetris. And what you're doing there is taking advantage of targeting of tumor cells versus normal cells from an antibody, which is binding to antigens on the tumor, so very selective, linked through to a cytotoxic payload. And in this particular case here, this is a maytansine derivative. And maytansine itself never really made it as a single-agent anti-cancer drug because it really had almost no therapeutic window. It was much, much too toxic. But now delivering it through something like this, it is actually now shows efficacy as an anti-cancer drug, particularly at the moment for treatment of this particular kind of metastatic breast cancer. Now what you have here is a situation where this thing, which is a cartoon of the antibody, is approximately 150 kilodalton, and the payload is maybe about 1 kilodalton, and maybe you have about 2 or 3 of them on it. So to develop some of these really very potent compounds as ADCs, you do not need much material at all. And in principle, you know, half a gram or a gram of these quite complex compounds are enough to, once you make an ADC, probably do phase 1 and maybe even early phase 2. Okay, so that's kind of a summary of where we'd like to go. This is sort of a summary, which many of you would appreciate already, so I feel I'm preaching to the converted, so natural products and drug discovery. But what I like to emphasize is total synthesis is an increasingly viable and realistic solution to the supply problem, even for complex natural product structures. And if you combine that with tumor targeting in the cancer area, you do not need much of these compounds. And maybe this is a way now of at least arguing for funding for natural product synthesis combined with drug discovery. So we'll see what happens. Okay, so this is pretty much at the end of lecture 1. So various people I have to thank. So this is my group probably about 2 years ago. And so I guess 2 people I'll thank here. Simon Williams, who's just finished applying in A and D. Sarah Fink, who's an American student who came to me from Williams College on a Herschel Smith scholarship. And having finished with me, Sarah's now gone to China and joined Biodura. And what I've put in this acknowledgement slide is the destination of the people who've gone through my group. So the large majority of them end up in pharmaceutical industry or chemical companies. A few of them end up in academic jobs. So Simon Blakey, who's at Emory as a professor, is actually one of the people in my group who started the aplonine project quite some years ago. Alison, who I mentioned earlier, Simon and Kenneth, who both worked on leodomethalide, Steve Dolby, who's at Merck. This guy here, somehow he got away. So he was an undergraduate project student in my group, and he's really very good. And academically, he was so good, he could get his own scholarship. But he didn't really know when he wanted to continue to do a PhD, so I let him go away and do something else for a while, until I found out that I wasn't going to get him back because he'd gone off to Italy, and I'm sure Gilbert Stork would approve of this, joined a particular automobile company that produces fast and I'm sure expensive sports cars, Ferrari. So David probably is doing very well there, but I'm sure he'd do so much better if he was doing a PhD in organic chemistry. Okay, 2 more. So earlier this year, I reached the grand old age of 60, and this was an opportunity to have a group reunion. And so many of my former students and postdocs came back to Cambridge, and we had a symposium, some nice meals, certainly quite a lot of alcohol was consumed as well. And so there's a few people I'd like to pick out in this slide. Simon Blakey, who's now at Emory. Rich Tillier, who's very high up in Merck, which could be useful. Jonathan Goodman, who's one of my first PhD students at Cambridge, is now a full professor. Kenneth here, who's just submitted his PhD thesis. Then tomorrow, I'm going to talk about some of the work of Ed Anderson here at the back on spirostralide. This guy here, well, he actually wasn't a member of my group. He came along as a guest. He's a good friend, but also allows me to show, I think, the next slide, because we like to combine having sort of reunions in Cambridge with some activities on the river. So in this particular case, Carl did handsomely. And fortunately, he could swim. We did worry a little bit. Okay, so let me finish up there. This reminds me to thank Gilbert again for endowing this lectureship series, which is very important for emphasizing the continuing importance of organic synthesis, be it to make natural products, complex natural products, functional materials, or really just anything. Synthesis is ultimately, well, inevitably, a creative science. Okay, so tomorrow, for those of you who want to come back and haven't had enough, I'll talk about our work on spirostrelide, so I'll finish there. Thank you very much.
We'll make an announcement before we take a few questions. Tomorrow after the lecture, there will be a reception in the Shane Atrium. And in case any of you failed to get all those structures drawn and those reagents transcribed, Ian has offered to leave a copy of his talk as a PDF file so that people can look at it. Of course, we also have the movie that you can view in your spare time. Questions?
So I forget the name of the compound with the NT structure.
Yeah, spangen.
Thank you. So I guess an obvious question, does the organism that produces that live in a cold, dark place?
Potentially it could, yes. But the mix of bacteria can sort of inhabit almost anywhere. And but one thing I should emphasize is that with these pentaenes, there's various other congeners. And the only one that was really isolated and properly characterized is the one that we synthesized. But it's kind of curious that it does produce something which chemically is not that stable and produces probably all possible permutations.
Do you think once one is made, then all isomers?
Well, I think that's the most probable, because certainly the one that we made seems to be the least stable, or one of the least stable possibilities.
Then it's curious if that was the one that was isolated and structurally characterized. But
well, sure, yeah.
And you get the cyclic reactions of the central segments all set up to
Yeah. Well, that's the other thing we have to avoid, as well as light, heat. So if it warms up at all, you start to degrade the pentaene. So they're really unfriendly compounds to work with.
Ian, you mentioned photosynthesis of protected natural clubs.
Yeah.
And as a member of that club, who has spored off with triisopropyl silyl ethers, what is your most unfavorite silyl ether when it failed to come off?
Well, this is very difficult to say because Gilbert introduced it. Occasionally TBS ethers can be a problem. And what is remarkable is in our work on rhizopodin, okay, so it's a symmetrical tetraol. If we had, so it has an oxazole, or two oxazoles, gem dimethyl. So the hydroxyl, which is in between the oxazole and the gem dimethyl, if that's a TBS ether, and if the other one, which is near the sort of dienoate of the macrocycle, if that's a TES ether, we could not convert that into rhizopodin. So what happens is where that TBS ether is really quite crowded, and what it prefers to do is just eliminate to give a double bond in conjugation with the oxazole. The one that did work, and this was at the third attempt, was to put a more labile silyl protecting group between the oxazole and the gem dimethyl, which was a TES. So a TES, and then the other way around, so it's kind of switching things over, was TBS ether on the other position, and that worked. So that was, of course, big relief, but that was at the third attempt. But what is worrying is in the literature, ahead of us, there's a claim of a total synthesis of rhizopodin. And what they make, and it's really quite inconclusive because there's no real characterization of the last two intermediates, and then sort of crude material is put on HPLC, and they isolate something like 140 micrograms. They claim that they can take tetra TBS rhizopodin and deprotect to give rhizopodin. So I certainly don't believe it. Yeah.
More questions? Student questions?
I just want to follow up briefly on Sam's question with this polyene that we were kind of answering a little bit back to the slides. This metathesis that comes
Yeah.
down, why ends up with that selectivity? You see early in the reaction, you see different products, and then it basically funnels its way back to that.
Okay. Well, I don't claim to have done that work. That was the isolation group. So what other things they isolated, I don't know. But what's in their paper, it's the only thing they fished out in 15% yield is that dyne. But almost certainly, yeah, they've got other things too.
This is from the ethylene.
Yeah. That's not our work. That's the isolation people. So.
When you were talking about the Zimmerman-Traxler transition state, you invoked a hydrogen bond from
Yeah.
high hydrogen.
Yeah.
Methoxy.
It was a PMB ether to the formyl hydrogen.
Right, so what's the evidence for that hydrogen bond?
Yeah. Okay, so experimental evidence is on the basis that if you analyze that transition state any other way, then it looks contrasteric because you're putting the bulky ether in the inside position, okay, to explain the stereochemical outcome, which is a fact. So my colleague at Cambridge, Jonathan Goodman, who's pretty much now a computational chemist, has done in-depth DFT calculations of that particular transition state, related transition states, and does very clearly find this sort of close connection between that ether oxygen and the proton of the aldehyde, suggesting, yes, a formyl hydrogen bond.
That's an interesting hypothesis.
Yeah. But another system, which is even more remarkable, is if you don't have, so at the moment in that system we have, alpha methyl, beta alkoxy, right? So the inducing stereocenter is the alpha methyl. If you remove that methyl group and move it over to where the ether oxygen is, so it's a beta stereocenter, and you do the same boron-aldol reaction, you actually end up with 1,5 stereoinduction in the antisense with very good selectivity, in many cases about 15 to 20 to 1. So this is totally remarkable in terms of acyclic stereocontrol. And again, the only way that I feel comfortable explaining it is that formyl hydrogen bond model. And again, the DFT calculations support that. And if you do things which are like interfering with the possibility of that hydrogen bond, for example, putting a bulky silyl ether group on it, then the selectivity goes down as you would expect. But of course, this was not planned. The great thing is, you discover these things and you try and figure out what the hell's going on.
All right, well, I hope you'll join me in thanking Ian for a great lecture, and we're looking forward to tomorrow's.
Well, nobody told me how long I should speak for.
Well, it's your choice.
Yeah. Well, not many people left, so that's a good sign.